Hello, I'm Rebecca Ash. This poster is on assessing whether species distribution models are reliable tools for understanding how living marine resources will respond to climate change. These models are very commonly used to project future distributions and sometimes abundance of different fishes, but they're assuming basically the relationships between fish and the environment are stationary and don't change over time. From the marine heat wave that hit the California current in the mid 2010s, we know that that assumption is violated. And the question that I'm trying to address here is, do we have other evidence of non-stationarity and changes in how fish relate to the environment in the past when looking at other regime shifts? So I studied this in the context of 4 small pelagic fish species. These include anchovy, sardine, jack mackerel, and chub mackerel. And I use generalized additive models here. We do have a second type of model that I examined, but that's not included in this poster. And we looked at basically changes in how fish relate to temperature, salinity, oxygen, and zooplankton volume. And this was done across several different types of regime shifts. So basically what I did is look at a model for one regime and then for another regime and evaluated whether you got the same model. If you do, then you have stationarity. If you don't get the same model, then you have evidence of non-stationarity. This was done for the Pacific decadal oscillation and also for change points in the time series of zooplankton volume and the spawning stock biomass of anchovy and sardine. So we have 3 different types of regime shifts based on kind of those criteria. And then I looked at stationarity across 6 different metrics. These are basically whether you have the same variables included in a model, whether fish relate to the variables in a linear or nonlinear fashion, what variables are the most important, the response curve shape of basically how fish relate to a given variable, the degree of responsiveness of a fish to that variable, and the preferred environmental range used by a fish. And this chart, which shows all those metrics, basically anything that's purple indicates that there's some non-stationarity between regimes. So you can see that for certain metrics, there's a lot of purple. Why does this matter? Well, this matters for our ability to make accurate future projections about what climate change might hold. So to demonstrate this, I made some projections for sardine using these different models developed across these past regimes. And basically what I found is that the variation between the different projections grows over time under more severe impacts of climate change. So this non-stationarity in how fish relate to the environment might actually become more important in the future and cause us to make potentially erroneous projections about how fish might respond. So with that, I'm going to end and see if people want to talk to me about this work later.
Hi, I'm Rebecca Howard, and I'm a PhD student at Oregon State University. This work is part of my master's thesis research and aims to examine how fishery-independent and dependent data collected on Oregon's commercial non-whiting groundfish fishery may complement each other for spatiotemporal analyses. Limitations on the temporal and spatial extent of fishery-independent data has led to knowledge gaps about the shelf fishery in particular, as NIM surveys have not been conducted for every year or season, and there is an absence of data inshore 55 meters depth. Fishery-dependent data collected by fishing vessels, logbooks, cover shallow waters and a broader timescale, but there are concerns about accuracy. Through this research, we found that in the 80s and 90s, the surveys have the most information gaps and highest potential to benefit from complementary use of logbook data. Commonly caught flatfishes, like Dover sole and petrale sole, seen here in the middle and right panels, had better spatial sampling coverage compared to shallow water species like starry flounder, depicted in the left panels. Overlap between datasets was variable, and lack of year-round sampling coverage may miss information about seasonally migratory species. This research provides context for future management of shelf groundfishes by illustrating where knowledge gaps lie in both data types and how they complement one another.
Hi, I'm Stephanie Jaeger, a marine scientist with the City of San Diego. I wanted to share a brief glimpse of results for mealtime moorings that we installed in partnership with Ube Sun's lab at Scripps. One main goal of this project is to better understand the dynamics of treated wastewater effluent from the city's 2 ocean outfalls and how the water quality and conditions change throughout the year. These moorings are really heavily outfitted with a suite of physical and biogeochemical sensors at multiple depths and have proved very useful for characterizing local conditions. For example, in Figure 2 on the left, we see at Point Loma at the mid-water depths at high salinity and low temperature waters, we see a characteristic signal from upwelling of low dissolved oxygen, and we can better understand the timing and influence of these events. Also, the large coastal dinoflagellate bloom that happened earlier this year in May was observed, and these data can help contribute to understanding the dynamics and impacts of these types of blooms. We're also in the process of identifying the best ways to catch signals from the wastewater plume. One example so far is from Point Loma. We see low salinity waters, likely from the influence of the plume on this plot. Of course, this is a big team effort to deploy and recover these moorings to quality control instruments and data, and this has been a great partnership between the city's Marine Biology and Ocean Operations Group and Uwe Sen's team at Scripps. Thank you for listening, and feel free to reach out to me with any questions on the project or if you'd like to access the data.
Hello, my name is Amanda Whitmire. I'm the head librarian at the Miller Library at Hopkins Marine Station of Stanford University. And this poster summarizes some work we've been doing to extract data from a 23-year CalCOFI time series in Monterey Bay that was undertaken between 1951 and 1974. After we had all of these materials digitized, we've been working through various processes to extract the data from the scans and make them available as spreadsheets so researchers can actually use it. This involved a combination of optical character recognition software, lots of student labor, and a crowdsourcing platform from the page. If you have any questions, shoot me an email or send me a tweet at my contact information at the bottom right of the slide. Thank you.
Hello, my name is Lainey Clunas. I am an undergraduate at CSU Monterey Bay and will be applying to graduate schools next fall. Today I'm going to be sharing my project regarding remote sensing of phytoplankton within the California current system. Prior studies have described a nutrient gradient between stronger coastal upwelling and weaker offshore wind stress-induced upwelling. This nutrient gradient causes a gradient between ecosystem regimes within phytoplankton community structures. Using in situ and remotely sensed data, our study aims to answer: how does phytoplankton biodiversity vary perpendicular to shore across a nutrient gradient from coastal upwelling to wind stress curl upwelling? And how well can remotely sensed images portray this phytoplankton size class distribution? We anticipate to see matchups between in situ and remotely sensed data supporting the phytoplankton size class gradient from onshore to offshore. Given that different phytoplankton size classes support different trophic systems, our results have the potential to hint at shifts within higher trophic levels.